Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, and 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us. The 163rd seminar, and I call it a JCVI day. For 1 month, I have prepared three proposals and written multiple papers. And tomorrow I will embark on my 15 days trip again to Europe to attend an international conference and give an invited talk as well as meet and catch up with many friends and colleagues. Despite all the challenges we face, international conferences and this seminar series must go on to solve many global problems together. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Sanjay Vashee. He is a professor and Rockville campus director at J. Craig Venter Institute or JCVI. I got to know him last year when I joined JCVI in May, and I really appreciate his kind acceptance of my seminar invitation. He is the second pioneer speaker from JCVI, and the first speaker was John Glass, who is also La Jolla campus director. And also, one interesting thing is Sanjay is from Zimbabwe, Africa, and also one of my best students from the same country. So that is another connection between him and myself. Sanjay, thank you so much for your great contributions to the scientific community, and the virtual podium is all yours now, and thank you so much.
Thank you, Taesuk. It's an honor and a pleasure to join this. I commend you on your on carrying out this process of trying to mentor young scientists and provide a platform for them to showcase their research. So as Taesuk said, we do have a connection. Not only do we work together, but his student is from Zimbabwe, and I was born and raised there. Before I moved, I started my college at the University of Illinois, where I got my BS. I was not a particularly strong student at the time, so I didn't know what I wanted to do. And so for me, taking a biochemistry lab class was the impetus. And this is when I was a senior that really got me interested in research. By then, it was obviously too late because my grades were not that great. So I had to go get a master's, where I went at Western Illinois University, where I got a master's in chemistry, and then did my PhD at the University of Texas in biochemistry. And then I was a postdoc at Johns Hopkins School of Medicine before I joined the Venter Institute, where I helped work on the synthetic cell. And so essentially using the tools that we developed or used for the synthetic cell, I then moved into what I call synthetic virology, where we're using synthetic biology to engineer mainly eukaryotic viruses in a more high throughput fashion, genome-wide, combinatorial style, so that we can accelerate therapeutics and vaccines. And we believe the approach can also work for bacteriophages, any phages, whether they're RNA or DNA. And particularly, we're much more interested in the non-model phage. But what I really want to talk about here are sort of lessons I learned as I was going through my career that are not related directly to your project or things, but the intangibles that I believe that the young scientists will need or key on to really succeed and progress in their career. So the first thing for me was not to key on techniques I learned while I was doing research, but how you approach the problem, the scientific problem. And so if you learn that, you will be able to move within fields, start new fields, able to do a lot of these things. And so key on that concept as you're learning as a young scientist. So what is the problem? What is known? What are the gaps? Do I need to develop technology? What are the big questions? Do I need to go outside my area to do all of the things? And so as you learn that and really hone that skill, I believe you'll be much more adaptable. So given how we're changing, the science is changing so fast, you'll be able to adapt. And of course, I believe the young rising star is within that realm, is already starting to think that way. So I commend him on that. So, but the other thing that for me was key that you never learn while you're a scientist is how important communication is. And how you have to learn to sell the science you have is, this is something that I realized when I was going through high school or even college was I didn't key on talking or writing. And it's just amazing how no matter what field you're in, how important those skills are if you can hone that. How do you sell anything? You need a salesperson in a business. You need to sell yourself to get hired as a professor, assistant professor, or the job. And then the other thing is, if you're in an academic setting, you need to be able to write well enough to sell your idea to people so when they review, they'll be able to understand what you're doing. Not only at the academic setting, even if you go to a company, you still have to sell that research project and how it's going to make business or etc. So key on these components because I myself wish I'd really done that a lot more than I did. And the last thing I want to say is network. When you go to meetings, go seek out the people that you really admire. Go talk to them because as much as your hard work is going to show what you do, other people will open doors for you. They will be able to, find opportunities or let you know about opportunities, and maybe potentially even hire you. So work on those networking skills as you're going. Those are the concepts that I think, if you look at and key on outside of your research, I think you will do well. With that, I'll stop on my soapbox and I'm excited to hear what Ben has to say. And again, thank you, Taesuk. This is such an awesome opportunity that you're providing for the whole younger scientific community.
Thank you so much, Sanjay. I mean, that is absolutely wonderful advice. I mean, that you cannot find from the literature. That is only you can gather, of course, by experience the most, but you could tell us earlier and then we can listen to earlier, that is much better. And also I also realize right now, my student, Tatenda Chopra, you also share the same nationality, born over, raised over there. At the same time, it might be coincidence, both of you working on virus, he developing vaccine, or also you're developing vaccine and working on virus. I'm talking about he working in Pfizer, but he also sort of adapt, as you mentioned. Adaptation is very important because he learned many things from my laboratory, but he only learned about RNA engineering or biochemical engineering, but somehow he never worked on the eukaryotic or mammalian system. But now he developing COVID vaccine, also developed already, COVID vaccine over there. So, he probably already knew what you just mentioned and then how to adapt to new environment, and then he's successful in that way. So I'm really appreciating. I mean, you just mentioned that one in that way. A lot of young people also adapt better. And then other two is absolutely important, networking and communication. Absolutely great advice. Thank you so much. All right. Um, Now, the main speaker of today with a longer introduction, Dr. Ben Adler is a postdoctoral researcher in the Jennifer Dona's lab at UC Berkeley, studying how diverse CRISPR-Cas biochemistry impact phage infection and how to leverage these activities to unravel the diverse genetic content they encode. Interestingly, today's topic is all about phage or virus. And also, as you know, virus is very well known for their adaptation. That's another, advice. I mean, Sanjay already mentioned. Ben obtained undergrad degree in bioengineering as well as biochemistry and cell biology at Rice University, followed by a PhD in bioengineering at UC Berkeley and UC San Francisco in the Adam Arkins lab. Professor Arkin is my academic grandpa, so technically Ben is my academic uncle in a sense. Outside the lab, Ben is a avid home cook, origamiist, and also chill pepper gardener. I want to see, or taste his, cooking, outcomes, sometime later. Ben, thank you so much for your time today, and please take it away, all yours now. Thank you.
Wonderful. Thank you for the incredibly kind introduction. I'll be shifting all the technology over to my screen. Let me see. So share this desktop, and is everything good on your end?
Perfect. Perfect.
All right. Hi everyone. My name is Benjamin Adler, and after that introduction, I guess I'd like to talk today about a lot of my work for my postdoctoral research on bacteriophage editing and functional genomics, and how selection is really all you need. Shown down here is an image I want some folks to think about today, about this antagonistic interaction between bacteriophages and their hosts. That is, bacteriophages are viruses of bacteria that have ultimately an incentive to be able to inhibit bacteriophage replication for their own survival. This is an innovation engine of sorts, and some of the things that have confounded technology development to date, but I want to show you guys here how we've gone about surpassing that. For the uninitiated, and for those who aren't bacteriophage lovers yet, bacteriophages are viruses that infect bacteria. And I'm going to show you an incredibly simple model of infection, but just please understand this is as simple as it gets. First, bacteriophages from the environment need to adsorb to the outside of a bacterial cell to be able to inject their genome, upon which they express genes that are important for taking over the cell and replicating, ultimately producing more virions than there were before, that ultimately need to get out of the cell and lyse. And this is as simple of a process as it can get, and this is ultimately many iterations on a theme. But what many folks don't know are that bacteriophages are the most abundant biological entities on Earth. That is, there are about 10 to the 31 virus-like particles on Earth. There are about 10 to the 31 virus-like particles on Earth. And maybe one way of kind of thinking about that is that for every grain of sand on Earth, there's about 1 trillion bacteriophages. And that model I mentioned earlier, this is really informed by our model phages. To this crowd, most people probably recognize these as the occasional 2-letter label they see on an enzyme tube for their favorite DNA ligase or RNA polymerase, but also maybe for the introductory gene circuit section with lambda. But what many folks don't realize about the model phages is only about 50% of their genes even have a predicted function. That is, despite being studied for over half a century, and we still only know about 50% of what they do. And as you can imagine, this is a very small piece of this very global piece of biology, and the non-model phages have only about 10 to 50% of genes of any known function. And as a synthetic biologist, maybe what I'd like to folks to think about here is that when we look at a model phage like T4, which has been studied since 1950, so over 75 years, is that this is an incredibly complex biological machine. That each of those very simple steps I mentioned earlier, these encode entire biosynthetic pathways to be able to shuttle energy into phage production as opposed to standard cell metabolism. They encode really complex protein components that can form this very finely crafted Moonlander-like virus. And they also encode genes to be able to subvert the host. And to me, what's really inspiring about this is that this is really a distilled series of selection pressures. For microbial ecology, this is known as the Red Queen hypothesis. That is, both sides of this antagonistic interaction have to be able to genetically innovate in order to survive. So, for instance, for these very basic phage functions, like the absorbing to the cell, being able to take over the cell, nonetheless replicating and lysis to get out of the cell, there's an implicit evolutionary pressure for the host to be able to simply evade phage predation. But they can also do other more complex strategies. So for instance, they can also rewire their own transcription to become less recognizable. They can also encode anti-phage immune systems. But even phages in the light of those challenges aren't captured without the ability to innovate. They can adopt new lifestyles, say, for instance, to compartmentalize their own genome in the middle of infection, or even be able to subvert said immune systems with, say, immune antagonists. Hosts are able to adapt to even those pressures. They're able to create adaptive immune systems like CRISPR that you'll be hearing about today. And maybe, controversially, they even have learned to differentiate in order to avoid phage threats. And what I hope people can see is that what is otherwise an incredibly simple picture actually reflects some of the most complex and diverse biology that there is. During my PhD at UC Berkeley in LBNL with Dr. Adam Arkin and Vivek Medalik, I used high throughput screening to be able to understand how phages interact with their host. This is a series of high throughput genetic screens that were performed in E. coli and Salmonella on the host side of the equation, where we knocked out every single gene and barcoded them so that we could track them and watch how they perform in response to phage threats. And while this was really informative as to what phages needed from the host in order to survive, this was fundamentally genetic information about the host. And whenever we were left with a question about phage biology, that is, what is important on the phage genome side of the equation, we were left looking at these genomes where the vast majority of genes were of unknown function and very few genes were of known function. And we were left without too many threads to be able to ask about what was going on the phage side of this ecological dynamic. So in 2021, I joined Dr. Jennifer Doudna's lab as a CRISPR pioneer, and I was hoping that we could really solve the phage side of this equation, or make a major inroad at least. And so I shifted to the phage side of this equation, really to get out of this idea of we are fundamentally limited by phage genetic dark matter. And I kind of came in with a singular goal of bringing phage genetics out of the dark ages. So over the course of my postdoc, I've developed robust tools for phage genetics that I'll be talking about today. We've been using some of those tools to be able to understand core features of phage infection. And also, I'm interested in using mechanisms of antiphage immunity as kind of a way of understanding what selection pressures are being imposed on the phage side of this equation. But for the purposes of today's talk, I'll be primarily focusing on the tools, but I'm more happy to talk with anyone about these other two bubbles at other points. So a very quick overview of what I'll be talking about today. I'll be talking about a pair of Cas13-based tools for phage genetics. The first of which is focusing on phage genome editing, on how using a kind of a surprising approach for phage genome editing by targeting phage RNA instead of its DNA, and this is true even the case for DNA phages, as well as a strategy that we called CRISPR-I-ART, or CRISPR-I through antisense RNA targeting, as a platform to use to perform phage functional genomics, where we use a catalytically deactivated version of Cas13d in order to knock down gene expression in phage genomes. And so beginning with phage genome editing. When starting in the Dana lab, I was thinking about these three core engineering goals for a modern phage genetics toolkit. Probably first and foremost, and the biggest challenge of this whole project is something that is effective across phage genetic diversity. When folks think about microbial genetic diversity or eukaryotic genetic diversity, people typically think about things in terms of phylogenetic trees. That is, you can track the hierarchy of how genetic information is inherited. And phages and eukaryotic viruses are actually a little different. They are, in fact, so diverse that you get these kind of islands of relation. That is, individual genes and proteins may be shared across genomes, but you won't actually be able to say very easily how closely individual phages are related to each other outside of these islands. And that has been a major challenge to be able to edit phage genomes writ large. I also was looking for tools that are scalable across the genome, that they're easily able to retarget individually new loci, as well as being easily programmable, say for instance a CRISPR guide RNA, and adaptable to genetic screens. And that way, in the perfect world, we can not only look at kind of host side diversity and be able to track barcodes, but also be able to track them in phages and being able to sequence them and figure out what genes are important and when they are important. And CRISPR seems like a pretty obvious solution to this. And I'm about to go into why that is not exactly the case. But in nature, CRISPR-Cas systems natively target invading phage genomes. This is kind of known as the primary adaptive immune system within bacteria. There are effectively three components that are worth thinking about. The first of which is the effector, and this shown here is FN-Cas12a. Which is the effector. This is the part that does the DNA damage in the context of phage infection. There is CRISPR adaptation systems. This is what incorporates memories from prior phage infection, as well as the CRISPR array. And these are the memories of prior phage infection events. The CRISPR array gets processed into a guide RNA, which then has memory of a phage genome that it had seen and needs to launch an immune response against. And this allows the effector to target the phage genome alongside a PAM, which is important for Cas12 binding to the genome. And then it can cut it and restrict the phage. And this has been tremendously helpful for genome editing in all sorts of organisms, from humans to bacteria. But one of the problems is that phage evolution fundamentally selects for escape from many of these common genetic tools. And over the last, basically since its discovery, there's been a lot of literature that has lamented how difficult it is to actually use CRISPR systems to target phage. And so obvious candidates of CRISPR effectors to use, say for instance Cas9 and Cas12, are actually relatively inefficient anti-phage immune systems. So we can't really benefit from these genome editing properties of them in the context of phage infection. And I'm going to list out a couple, but not exhaustive examples of why it is these are difficult to use. Phages can encode their DNA or encode their genome in several classes, ranging from double-stranded DNA to single-stranded DNA and single-stranded RNA, of which Cas9 and Cas12 can only target two of these. Some phages, like for instance, phage T4, can have really dramatic DNA modifications. In the case of T4, it glucosylates with alpha and beta linkages, all cytosines on this genome. Some phages, like phage Gosler, are able to compartmentalize their genome and form a subcellular structure. And this is in the sense of phage-encoded nucleus, in which case such CRISPR effectors cannot even access the phage genome during infection. And then it's one kind of feature, maybe bug of it all, is that a lot of phages, especially the DNA phages, have established these really intricate DNA repair systems that allow them to deal with being cut once or twice on their genome. And this makes it very difficult to use CRISPR systems as kind of a modular tool for restricting phage infection. But when I was joining the lab, it was becoming clear that CRISPR diversity was really extending well beyond Cas9 and Cas12. And when I was joining the Dana lab, I was curious about where other CRISPR effectors kind of fell in this tree of phage restriction. So shown up here is a very simple model of a molecular dogma view of phage infection, that is for the double-stranded DNA phages at least. Their genome is encoded in DNA, which after it injects, it circularizes. Genes are transcribed into RNA, which is translated into proteins. And after kind of this nice ordering of expression of all of its genes, you ultimately get viral replication and virus production. And if in this kind of molecular view of phage infection, this is where Cas9 and Cas12a would target. They just cut the DNA. But early on in my postdoc, I was working with a really talented grad student, Carolyn, now Dr. Carolyn Wang, on a CRISPR effector known as Cas12c2, which surprisingly didn't actually cut DNA. In fact, it actually just blocked transcription, much like we would think of with CRISPR-I. And that was sufficient for at least a few phages to restrict infection. Also, but the focus of my postdoc work was focusing on a CRISPR effector known as Cas13, which was originally thought to target the invading RNA of RNA phages. But over time, it became pretty clear through my work and others that targeting RNA was actually targeting this kind of universally conserved molecule over the course of phage infection that was capable of not just in stopping RNA phages, but actually DNA phages as well. And this is going to be the star of the show today. So if you remember anything about Cas13, just remember the word RNA. It is an RNA-guarded, RNA-targeting, RNA-cleaving CRISPR-Cas effector. Cas13, using two catalytic domains on the outside, on that kind of the outside of the CRISPR effector, known as HepN, uses its guide RNA to target an invading phage transcript, and upon recognizing it, the two HepN domains are brought closer together, forming a complete active site, leading to RNA cleavage of not just the targeted transcript, but also all RNA in the cell. As you can imagine, for a nascent viral infection, this is a bit of a catastrophe, and the cell goes into dormancy. The cell goes somewhat dormant, and thus ultimately gets to phage restriction. But when I was starting my postdoc, it was actually unclear if this model really mattered for DNA phages. So in my first experiment, and so shown here is kind of my first experiment in the lab. Alongside another postdoc in the lab, we built a series of Cas13 expression vectors where we have Cas13 under the PTEP promoter, which is an ATC-inducible promoter, as well as a very simple programmable CRISPR RNA using a single repeat and a spacer. In this case, this is a model of T4 infection where we are using a Cas13 effector to target the orange gene. And the model is if Cas13 is able to target that RNA, and whether or not that cleavage leads to DNA T4 restriction. And so shown here is an example of what this type of data looks like. Most of the data a lot of the data I'm showing here today is ultimately the result of this style of an assay, which is known as a plaque assay. What this is a lawn of bacteria, and these dark dots over here are individual infection centers from an individual phage infection event that is cascaded out into a plaque or a hole in that kind of lawn of bacteria. You can kind of think of this as a colony. And typically this is done through serial dilutions, and then you count the plaques and you get a sense of titer or how many phages there were. So when you were using a guide RNA targeting RFP, so basically a target that was never present in the experiment, you get no phage restriction. But if you target, say, an important gene in phage T4, in this case, the major capsid protein, you get 10,000 or 100,000-fold restriction of phage T4. And while you're left with some clearings at the top, you don't see any of these really infectious centers or plaques. And you can turn this into an efficiency of plaquing calculation by simply dividing the estimated titer of the phage in the negative control condition over the number of infectious centers you see, in this case approximated as 1, where in the case where you're actually targeting the phage genome, and you can see that you get about 100,000-fold restriction of phage T4 when you target it versus when you don't. We next wanted to see whether or not this style of targeting was effective across the T4 genome. And so to do this, we pecked a series of known important genes in T4s, ranging from the major capsid protein to DNA modification proteins, to important regulators within T4, inside the coding sequence, outside the coding sequence, and even within the coding sequence. In addition, we also wanted to see whether or not genes that were non-essential, that is, genes that are not necessarily important for phage infection, at least in standard laboratory conditions, would also be restricted by Cas13. And broadly speaking, what we found is it didn't really matter what we targeted as long as we targeted it. As long as we did so, we would get about 10,000-fold restriction of phage T4, suggesting that Cas13a is a versatile and potent anti-phage restriction system across the T4 genome. I next collaborated with basically with a postdoctoral researcher in Jill Banfield's lab, Dr. Thomas Hessler, who at the time was playing around with how to phylogenize phages across metagenomes. So what we did is we mapped all E. coli phage genetic diversity and tried to map onto it the phages that I knew I had in the lab, which are kind of shown here in black. The E. coli phages are shown in red, and related but not obviously E. coli phages are shown here in blue. What we did is we designed two CRISPR RNAs across either early genes, so genes that are important for establishing infection, like an RNA or DNA polymerase, as well as a guide RNA targeting the major capsid protein, which is super easy to identify in phage genomes. And then we tested Cas13 against all these phages. And what we see over here is that we get results that look almost identical to phage T4, where when you don't target the phage, as shown as the left in all 8 of these panels, that you have relatively robust phage infection, but when you target either its DNA polymerase or major capsid protein, that you always see restriction of a very strong restriction of the phage by at least 10,000-fold. And what this suggested to us is that Cas13 efficacy was both unaffected by target phage, it was unaffected by gene expression timing, and that maybe in a sense of a failed experiment is actually a successful experiment. Phage-encoded mechanisms for Cas13a resistance are relatively rare, or at least in E. coli. And one thing that I realized when we saw this very core result was that we immediately had a major success across this effective across phage genetic diversity criteria. Because it's a CRISPR system, we knew this is scalable across the genome and easily adaptable, programmable, and adaptable to screens. What I didn't tell people in this room beforehand is that the challenge with phage genome editing is not actually the ability to edit the phage. In fact, actually, phages are incredibly good at taking up foreign DNA. And this has been known since at least the 70s and 80s. Where if you have a plasmid that has homology arms, say for instance 250 base pairs of phage genetic DNA surrounding an edit of interest, in this case this little purple sliver over here, you can at some rate, which is typically pretty low, about 0.01 to 1% of infections kind of leave with this result. You can convert the orange gene to a purple gene and thus have a very low fraction of edited phages. What has been the problem since the 70s and 80s is you typically need either a really useful screen to be able to do this, say for instance, either phage display or the ability to pick plaques and then PCR very carefully. And you just never really had an efficient selection to be able to do this. And that's where Cas13 falls into this process. So we proposed that using Cas13, we can do a very simple and efficient enrichment process. That is, we can use a very low multiplicity of infection. That is, though most cells never see an infection, at least in the first round of infection, but over time it can cascade. And all wild-type phages present in this experiment will get restricted by Cas13, but the edited phages, it will just look like the negative control. Cas13 won't be able to target the purple phages. And thus, we expect when we infect with this mixed population that we would get primarily the purple population out. And so shown here is what this actually is looking like at a DNA level. So shown here is a known dispensable gene in bacteriophage T4. This is SOC. We can design we can design more or less infinite guide RNAs against this gene, but in this case, we are just using two guide RNAs, guide RNase 1 and guide RNA 2. Using homologous recombination, we are designing a very clean deletion of the SOC gene using these two purple boxed homologous recombination regions. Using these two guide RNAs, we can both we can both enrich and also somewhat measure the efficiency of this edit being incorporated. So after just homologous recombination, you can see that only about 0.01 to 1.1% of phages are actually edited in this experiment. When you target the major capsid protein, which is never edited in this experiment, you get complete restriction. And when you don't target the phage, you see indeed no restriction. However, after using one of these guides, we can see that we get a edited phage enrichment to near 100%. This result was not just limited to T4. We also extended this to bacteriophage T7, as well as a phage that, to the best of our knowledge, had never been edited before, or a relative had never been edited before. In all cases, you get about 0.1 to 1% of phages getting edited using just homologous recombination alone. But after selection with Cas13, we could always get this to near 100% for at least gene deletions. We also highlighted that if you design your homologous recombination arms differently, you can do multi-gene deletions. So shown here is a case with T4 where we deleted nearly 10 kb of its 168 kilobase genome. And you can also use recoding to be able to recode a region of interest. In this case, it allows for very minimal edits that can go down as small as down to a single codon that deviates from the wild type sequence. And this is all selectable using the exact same CRISPR-Cas13 method. And again, these very tiny one codon edits are still able to go nearly to 100% efficiency after selection. So what we showed in this study is that using a single CRISPR-based method that was not really limited by whichever phage we wanted to throw at it, you can select for a wide variety of edits, ranging from individual codons to being able to delete individual genes. And while we didn't show it in this paper, we have since done this, and so have others. You can also use it to select for gene knock-ins as well. But also you can do genome scale level edits where you can fundamentally resize the phage genome using this style of selection. And this is not just limited to model E. coli. Colleagues over at the Weizmann Institute in Israel have shown that this method is very easily extensible to Bacillus subtilis phages, and colleagues down at UCSD have also shown that this is very easily extensible to Pseudomonas aeruginosa. And in a corroborating paper, a co-submitted paper with Dr. Joe Bondi-Denamy's group over at UCSS, has also seen very similar results using a different Cas13 effector as well as its anti-CRISPR selection marker. So in the last 5-10 minutes or so, I'm going to be talking about our work on phage functional genomics using a catalytically deactivated version of Cas13. One way of reframing the prior result is that RNA is a broadly targetable molecule during phage infection. That is, despite all of these different challenges phages can throw at a CRISPR system, RNA is always targetable during infection. This is some work that has been co-led with a grad student who now officially has his PhD, Dr. Muntathar Al-Shamari, as well as a then postdoc, now PI at the IGI, Dr. Brady Kress, where they observed that if you get rid of the catalytic activity of the hep-N domains of some Cas13 effectors, that you can not just target phage transcripts, but you can actually block translation of individual genes. And so I ended up joining this project asking a very fundamental question of asking if CRISPR-I through antisense RNA targeting, or CRISPR-I-ART, can discriminate gene importance in phages. That is, in the case where we have a non-essential gene, so like SOC in the earlier example, do you see robust plaque formation such as this? Or when you target a essential gene, say for instance the major capsid protein, do you see a reduction in plaque size, as shown down here, and or reduction in titer? And broadly speaking, that is what we saw, that if you targeted an essential gene, you would always get a drop in efficiency of plaquing, or a reduction in plaque size. Whereas if you targeted a non-essential gene, it's as if you never targeted the phage at all. This is not true with this is not something that you see with a lot of other CRISPR-Cas systems. For instance, if you use dCas12a, you see a minor phenotype with major capsid protein, and then you miss these other essential genes. And if you use dCas9, it just doesn't seem to work much at all. When you test it again, challenge it with phage diversity, you can see for many, but maybe not all phages, that this is a very effective way of knocking down efficiency of plaquing. But even in the cases where you don't see a drop in efficiency of plaquing, you do see a corresponding reduction in plaque area that might correspond to a reduction in phage production when targeted. And we did find that this was able to be extended across most, if not all, of phage diversity. Because it's a guide RNA, we can multiplex to be able to augment some inefficient CRISPR RNA phenotypes. So shown over here is a case with T7, which has these very characteristic big plaques. And when you target its RNA polymerase, you do see a reduction in size, but you don't see complete elimination of infection. And similarly, when you target the major capsid protein, there's a much more obvious reduction in phage plaquing, but not complete. But because this is ultimately a CRISPR RNA, we can design them as an array. And when you do this, you actually see near complete reduction of T7 infection. So the thing I was particularly excited about here is the ability to turn this into a transcriptome-wide gene fitness screen. So broadly speaking, the idea here is that basically the number of viruses produced is inversely proportional to the fitness of the gene that is targeted with the CRISPR RNA. Using twist DNA synthesis, we could design an oligo pool of guide RNAs, 7 of them per coding sequence. We could transform them into E. coli and then expose that library to phage or not to phage. And we played out all survivors. We can effectively mini prep them, and then send them out for sequencing using Illumina sequencing technologies. In order to determine phage gene fitness, I wrote a very simple pipeline to be able to count the guide RNA spacers, normalize them across sequencing runs, and calculate out a guide RNA fitness, which has relatively high, basically high experiment-to-experiment correlation. And then we would merge the guide fitness into a gene fitness through a very simple arithmetic mean. And then as a p-like statistic of spread, we would use a KS statistic. And this is a very complex view of what the type of data we were seeing. But fundamentally, what we're doing is we're looking at every single gene inside of a phage genome in a single experiment and being able to assess if it is important or not. And this is something that had never been done before in phage biology. So shown here is a volcano plot of basically how important phage genes were in phage T4. A lot of genes that were known were particularly important, and because phage T4 in particular has been studied for over 75 years, this is something that has been very carefully studied. And the vast majority of our hits that had high fitness were either essential, and some of the exception cases we actually found out were in ultimate agreement and mostly just hadn't been deleted before, or hadn't been attempted to be deleted before. This is extensible to other model phages such as T5, where we found the pre-early genes were especially important, as well as completely non-model phages like SOSP1 or PTX-U04. One of my favorite findings from this was that biological dark matter turned out to be incredibly important for phage infection, and this is especially important for non-model phages. So shown here is a very simple distribution of basically every gene that we called fit in these experiments, and whether or not it had an estimated known function, as shown in red, kind of an ambiguous function, say for instance endonuclease, where endonuclease of what we don't know, but it was an endonuclease, or a completely unknown function as shown in dark blue over here. And for some phages, say for instance PTX-U04, which is a phage that I think has popped up in one, maybe two other studies, you can have these genes that are completely of unknown function, but very dramatic. So for instance, this gene is about 10% of the entire phage's genome. It looks like a polyprotein if you alpha-fold it, but it's entirely of completely unknown function, yet undeniably important for PTX-U04 infection. And when we're kind of faced with these, the kind of parting thought I'd like to leave people with is that dark matter in phage infection is inevitably important for biotechnology. Some of the fundamental discoveries in phage biology have been the basis of what we understand about DNA and RNA replication, as well as DNA repair. The components for them, a lot of these genes are really important for variant and complex macromolecular assembly, or even kind of inspiring understandings of phage lifestyles. They encode genes that are important for offense against bacterial hosts that might be potential protein therapeutics, as well as kind of interesting stories in ecologies where they kind of often can offer gifts to the host to allow them to establish infection. They also encode important anti-immunity genes that also might be interesting as potential immunological type of therapeutic diseases. But zooming out a little bit, these are the basis for some of the fundamental nucleic acid tools that we use today. Thinking forward a little bit, these can also be really important components of how we think about mobilizing genes in complex environments and microbiomes, as well as manipulating and destroying microbes that we don't want. I have a lot of folks to thank for the type of science I've shown today. Probably first and foremost is my postdoc advisor, Professor Jennifer Doudna, as well as co-leads on some of the work I showed with CRISPR-IR, Dr. Muntathar Shmari and Dr. Brady Kress, as well as colleagues, Dr. Thomas Hessler, Emily Armbruster, and Julia Zhao. I also would like to give a special thanks to my undergrads, in particular, Alushi Lahiri, who's now a PhD student over at HMS. And while I didn't show any data that Kendall had collected, I'd also like to send a special thanks to Kendall Seh, who has also been a delight to work with. I'd also like to thank collaborators that have contributed to work I've shown today, Professor Jillian Banfield over at Berkeley, Joe Pogliano down at UCSD, and Rodolphe Barangu over at NC State University. If folks would like to reach out, my email is down below, as well as my Twitter and Blue Sky handles. And thank you.
Fantastic talk. Thank you so much. This is absolutely wonderful. Let me ask Sanjay whether he has question or comment.
Yeah, great talk, Ben. I'm intrigued a little bit between the two studies here because maybe I'm mistaken. So you said if you target non-essential genes with your cleavage, you were able to restrict infection, whereas if you knock down non-essential, you were able to not have any issues. So is there, if I'm not mistaken, can you explain why going after non-essential would also restrict?
So I think one of the ways, in the longer form of this talk, I like to think about one of the things I like to think about a lot is when is it important to target something that is non-essential? So in this case, when we have catalytically active, I like to think of this as you're tilting the ecological scales in a way of just saying, is this there or is this not? And if it is there, you destroy everything. If it is not there, you let the Cap13 ends up letting the phage live. In the case of CRISPR-IR, you're getting a fundamental readout on, given that something is there, how important is it or not? So in the case where we did end up having some genes that are in fact non-essential and indeed giving a important fitness result. That is, if you tried to delete them, you'd end up seeing a small and clear limit or limitation of phage replication. In this case, one example in the case of T4, I highlighted it up here, is DMD. So this gene over here is a known non-essential gene in T4, but what this gene is it is an antitoxin to a toxin-antitoxin system that is present on the E. coli genome. So while it is technically dispensable, the phage is definitely worse off without it. And so you can definitely see you can imagine that that leads to a true fitness result. And so that is partially why you will have some genes that are known to be non-essential, but are still highly important for T4 infection. There are indeed some genes that we thought were essential that are not important for phage infection, and that might be somewhat technological. In some cases, it's just by design. So because we're effectively targeting the ribosome binding site with this technique, if a phage gene has multiple independent ribosome binding sites, you can imagine that it does create problems for this. And indeed, in this case, the T4 terminase is a case that looks exactly like this. It has 5 known ribosome binding sites that have been carefully studied over 75 years. In that case, we aren't able to get a phenotype against the terminase, despite it being obviously essential. But one of the things that I've found to be kind of this is a feature, not a bug about it, is that capturing some of these intermediate phenotypes where it is a non-essential gene, but it is undeniably important, is an incredibly important foothold to being able to ask questions about when something is important.
Great. And then a follow-up. So a lot of what you talked about is lytic phage. How complicated would you get now? There's a number of temperate phages, so that goes into lysogeny. And so how do you, is there a way your techniques can help tackle some of these phages?
Yeah. So I would say that one of the phages that we talked about today, SUSP1, is increasing. So one little bit of footnote is over the course of my postdoc, SUSP1 has increasingly been viewed as a not entirely lytic phage, in a way that people hadn't really predicted prior to the study. It's looking like it maintains itself as an episome, but you can find it in a recent report from, I believe, Andy Millard's group. Basically found that if you look at whole genome sequences of a lot of E. coli isolates, you find phages that are very closely related to SUSP1. So I do think this does work for at least the episomal phages. For temperate phages that say are maintained as prophages in genomes, this is actually something where I'm not confident that the editing problem is really a problem because you're mostly tethered to the ability to edit the host organism. So if you can do that efficiently, you can edit the phage efficiently. The lytic phage challenge was something where I was noticing at the beginning of my postdoc, I was noticing major gaps in the phage editing literature on this. That it felt like people were primarily either focused on T7 as the monolytic phage and just trying to mutagenize tail fibers, or really focused on lambda and maintaining as a prophage and editing it that way. And so I was trying to hit this missing need of being able to tackle lytic phages, in which case this series of techniques worked beautifully. And so I haven't really questioned that too much.
Thank you.
But I do believe that the temperate phage problem is maybe even a simpler problem than this in reality, where like I believe that if you can edit the prophages, you effectively have edited your lytic phage if you can get the prophage to pop out.
Okay, thanks.
So Sanjay, do you have any other question?
No, I think this is great. Thank you.
Sure. Let me ask one question to both of you that actually kind of bothered me for many years. But now I have two virus experts I can ask that question. So this is a little bit philosophical uh, or challenging question. So if you don't have answer, I'm okay, but I just want to hear your perspective. So given my very minimum knowledge of phage or virus knowledge, uh, I just kind of keep wondering where this virus or phage comes from. I'm talking about billions years ago. To me, based on all the machinery that is basically hijacked by virus, that is actually, in the cell, my assumption was virus or phage coming from the host, originated from the host rather than coming from outside. That is my kind of assumption, but I never, search for the literature deep enough. But I just want to hear from you where that guy comes from and then hurting us. It makes sense. If that's originally from host, like a cancer, that makes sense. We might generating some bad thing because all we have is both good side and bad side, and then somehow that hurting us or punish us. But I don't know. Where that comes from.
Okay, you were asked a really hard question, so I'm going to defer to Ben first, and then I'll take a crack at it.
Yeah, yeah, I agree. That's a really hard question. I'm not sure I can answer where they come from, but I definitely, one philosophical point, I feel like I haven't had too many opportunities to speak about a whole lot, is that I'm actually not super convinced that evolutionarily speaking, phages and eukaryotic viruses are that different. Mostly from the perspective of this has been known for the thing I'm about to say has been known for a while in a couple limited cases. Say, for instance, the major capsid gene. If you look at their protein structures, for instance, between between herpes virus and even the T4 major capsid gene, you can see that protein fold wise, they clearly are coming from the same place.
Yeah.
This all being said, the clear disclaimer is that there's no known phage, to the best of my knowledge, that has directly led to a human infection. There are maybe phages that have some intermediate effect through kind of other pathogenic interactions, but phages themselves are not known to interface with human infection. On incredibly distant evolutionary timescales, though, there are a lot of genes that are shared, and especially in an era where alpha folding genes are allowing you to kind of see through the wormhole of distant evolutionary like co-function. You can definitely see that a lot of the same threats that eukaryotic viruses face by, say, our own immune system, phages can see through the bacterial immune system. And in some very, very, very rare cases, you can kind of see these very parallel evolutionary trajectory and these viruses reaching the same conclusion. And there's some really beautiful studies that are coming out from basically colleagues of mine work over at the Gladstone Institute, as well as a series of phage immunity papers from Phil Cranches, Rotem Sorek, and others kind of on this topic.
So actually, Ben, you raise a great point. So herpes viruses, especially HSV1, has many, many things that it shares with phage biology, even how it replicates, concatemers, you name it. So I honestly think that a lot of eukaryotic viruses actually probably came from bacteriophages. And maybe it was intracellular pathogens, et cetera, that allowed exposure to this. But this is my personal bias. This is kind of thinking there's no real evidence other than, as you say, there's a lot of parallels between the structure shared in terms of the capsid. But there's a lot of mobile elements that you see around there. And so I'm wondering whether it was an offshoot of mobile DNAs going around that started building, providing a membrane around it that then allowed it. So, you raise a big, important question, Taesuk. And so, yeah, I don't know that I have an answer for you, but to me, I likely think they may have come from plasmids that have then, put some membranes around themselves, or mobile elements that popped out in and out, ice elements, all of these things that might have started that whole process. But that's an open question. Interesting. Never really considered it as much in terms of the evolution, and I should.
Yeah, so that makes sense. But the reason I'm asking that question is also from the practical perspective, because I mean the my assumption was, let's say billions years ago bacteria somehow appear on Earth, and then bacteria have some, mobile element, including plasmid you just mentioned. That's something I kind of thinking about. And then sometimes somehow plasmid become bad guy, kind of getting all kind of bad thing, and then just lyse the cell, and then they kind of that plasmid just doing that business for billion years, somehow. Uh, That is the only explanation I could come up with, because I mean I cannot think of some random event molecule just appear outside and then just infect our cell. That's not possible. The reason I'm asking that question is we understand that process, the origin of the, phage or virus. We could have the better solution rather than keep fighting against phage or virus. And
Yeah,
then, but I just
I
want
think
to,
you raise important questions in terms of just understanding the basic biology of these
yeah.
things. And again, as simple as they seem to be viruses, they're also very complex,
Yeah.
as you can see. And so
Yeah.
understanding them, I think, is very important. Hopefully, there's still that commitment everywhere to do basic research like this.
Mm-hmm. Absolutely. So now I see the 6 minutes after o'clock, so I probably going to close. So wonderful talk, both of you. Thank you so much. And thank you all for joining and staying today. We'll meet again on June 12th, Thursday, the same time, the same Zoom link. We'll have Dr. Eric Pierce Toff at National Science Foundation and Professor Claire McWhite at University of Arizona. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thank you again. I stop recording. Just 1 second.